All right, guys, just trying to make sure that we are recording. Recording, yes. Okay. All right, guys, so I wanted to start chapter 3. Chapter 2 is on chemistry. So I put a bunch of videos on there. There's those workout sheets. You can read that. I'm not super concerned about chemistry, but you will need to know ionic bonds, covalent bonds, hydrogen bonds. We're going to start breaking down DNA and how the nucleotides are all attached and things like that. You should have a general knowledge of that. This is not a chemistry course. It's not even a biochem course. But I'm assuming, I'm hoping that if you guys are taking this course that you had, at bare minimum, a good biology course in high school, and you took a survey of anatomy, physiology, or a human bio course or something. Hopefully you're not just randomly fast tracking this to get into whatever program. It can be done, but it's going to be a tremendous amount of work. I'm going to warn you guys again, and when I say guys, I mean ladies and gentlemen. It's just kind of a colloquialism I have. But if you're going to be taking this course, and I don't care how smart you are, a lot of you are probably smarter than I am, but you're literally going to have to spend 10 or 12 hours a week, especially if you're doing lecture and lab. So the thing is, with lecture and lab, they should kind of come together, and there's going to be a lot of duplication. So before I get started, for this course, any of the mastering activities are in this course as practice. You will do better on the exams and you'll get a better grasp of it. But for lab, it's mandatory. So if you want to do it in lab and not in lecture, that's fine. Or if you want to do it twice, that's even better. But some of these lab activities done properly are going to take 2 or 3 hours, which you can probably complain about all you want. But if you were literally here, you'd have to drive to campus and do all this, and the labs are 3 hours long anyway, so you can kind of do it intermittently in chunks. I'm gonna work out the details on that. So most of you got homework 1 or homework 2 into me through the Blackboard LMS learning management system. Some of you emailed it to me or scanned it to me. That's fine. I can manually put the grades in, not a big deal. But from now on, I think due to what's going on, I think we're going to do all of this in mastering. We have the technology to do it, so we'll do that. But let me just get started here. So I can record this. I'll put this in lecture and lab for everyone so you can access it. Some of you are in lecture, not in lab. Some of you are in lab, not in lecture. Hopefully you didn't pick one teacher for lecture and one different one for lab. It can be done, but usually it's not quite as cohesive if you do it that way, but it obviously can be done. All right, so I got a new toy here. Hopefully I can draw on this, or I can drop and drag using this thing here, but we'll see if it works. So we want to start talking about cell structure, and then we'll get into some tissue. So cell is basically the smallest biological unit of the body, and we're not getting into physiology quite yet, and we're not getting into pathophysiology yet, but please realize your cells are form, follows, functions. They're all going to be slightly different, whether they're muscle cells or cartilage cells or adipose cells or whatever they are. They all have a very specific purpose in the body. They are going to have very specific structure, and they're all going to have very specific things in their cytoplasm. All right, depending on what that job of that cell is. And we're also going to talk about a lot of these proteins, or the outside layer of these cells, and the integral proteins and glycoproteins, and how these are markers, and they can be docking stations for enzymes, and we'll get into some of that. So hopefully you guys will find this fascinating. It's going to be, like I said, it's going to be a whole new language. It's going to be, hopefully it won't be completely foreign to you, but even if it is foreign to you, this course, like I said, is the building block of hopefully everything in your future. So we kind of want to make sure we have this stuff down. And once you start learning how cells work, if you take me for pathophys or microbiology, we'll start talking about how these pharmaceuticals work, and how we can cause change in cells, or how we can We can fight cancer. We can do this just by using the markers in the cell to get some of these physiological agents into the cell and start altering their mechanics using that. So we could potentially, you know, give a pharmaceutical that is going to mimic an enzyme or a substrate or something, so we can cause a physiological change inside the cell and change the physiology and hopefully relieve symptoms or alleviate a disease process through the epidemiology of the disease process. Okay. Oh, so this thing is working. Cool. All right. So you guys want to watch the video. They're going to give you, you know, why it's important or how it relates to going and click on that. It'll give you how it relates to the field. So like, well, why are we learning this? Or, you know, it's not like we are trying to play stump the student and give you this. We just want to figure out, well, this is where it may come into play. And like I said before, we have to learn the alphabet, words, sentences, we do paragraphs and chapters. You know, that's how I want you to start thinking. This stuff is the minutia. We have to learn this. We can't build from this. Okay. So, like I said earlier, the cells are the smallest living unit. So there's over 250 different cell types in the body. All right. And like I said, we break it down into white blood cells, red blood cells, T cells, adipose cells, cardiac cells, smooth muscle cells, you know, myocardium, neural cells, neuroglial cells. There's trillions of different things we could give you examples of. So types are different size, shape, and subcellular components. These differences lead to difference in function. So remember, form follows functions. And each one of these cells, depending on if it's two different cell types, makes a tissue, and different tissue types make an organ. But each cell is going to have a very specific function. So therefore, inside that cell is going to have different organelles, and those organelles are going to determine what the cell makes, produces, or exports, or imports, or stores, or whatever. So this is A&P1. We're going to get some basics, but please realize it can get a lot more complicated than what's here. So, you know, you may think this is super complicated, but it can get even more complicated. Like working this pad thing. Okay, so here's different types of cells. So we have, you know, up here we have our fibroblasts. All right, so these are going to be part of our connective tissue, and these are kind of going to be for structure, or talking about fibrogen cells. They can help us with blood clotting. Here's our erythrocytes, or red blood cells. They're going to carry, but right now it's just, they're going to carry oxygen and carbon dioxide back and forth to the lungs, so we can oxygenate tissue, so we can run the Krebs cycle to produce energy, to keep our cells alive and be able to go through mitotic changes and growth. Here's skeletal muscle cell. Contractile, we'll start talking about the cross bridges and things like that later on, how these contract, and we can send messages down the sarcolemma to turn these muscle cells on and off. Smooth muscles are going to be a different spindle shape, and these are going to be for peristalsis in your digestive system, causing, you know, things to move hopefully in the right direction. Or we'll learn that these are going to be able to contract and constrict the diameter of the lumen of your capillary beds or your respiratory tract. And then when they relax, they're going to open fully, and then we're going to dilate these lumens or these channels to decrease pressure or increase blood flow or airflow or whatever we need to do. All right. Here's adipose or lipid cells or fat cells. You're going to basically just store energy for the most part, but we're also going to find out that some of these cells are around your kidneys and in the back of your eye socket, and they're going to be, always start thinking of them as bubble wrap. They're going to be there for protection and cushioning. All right, so please realize your kidneys are, you know, on the posterior of your body below your rib cage, and you know, in the upper flank, if you get hit or in a car accident or whatever, and they're not protected, they can be damaged. So we kind of want to have a little bit of adipose or cushioning to take any blunt force to those organs, and it kind of holds things in place too. All right. Here's a macrophage. Macro meaning big, phage means eat. So these are macrophages. These are going to go around your tissues of your body. They're going to start engulfing, cleaning up any debris, antigen antibody complexes, any dead cells, any kind of debris, or so just kind of think of them as street cleaners or tiny vacuums or things that pick up garbage, or when you repair tissue, that you go in and clean up all the debris, you know, and we'll get into that later. Here's your nerve cell. Please realize that this is the general nerve site with axon, dendrites, and propagation going in one direction, but they don't all look like that. So they're going to be different. That's just kind of a general figure of a nerve cell. And here's cells of reproduction. So a sperm cell, the only cell in the human body that has flagella. If you guys take micro with me, or Dr. Rowe or Dr. Hoffman or Dr. Bailey or whatever, we'll be talking about how some of these bacteria have flagella, which just allows them to move, or they have motility. All right, so generalization of cells. Remember, this is all general, and each one is specific in its own unique way, just like each one of you is. Generally, we can give you all the human characteristics or whatever, but each one of you has very specific talents or you know, attributes that are specific to you. But in general, we'll talk about some of these. All cells have some common structures and functions. And so all cells have a biphospholipid layer, and we'll talk about that. So the outer layer, it's key to all cells. It allows things in and out. So that's going to be a very, very key component. Sort of like your skin on your body. It allows certain things in, keeps most things out. Human cells have three basic parts. So the plasma membrane is a flexible outer boundary. We'll talk about that. It's a biphospholipid layer, so it's two layers, water-loving and water-fearing. One's going to be facing the outer or external environment, which is aqueous or fluid, and the other part's going to be, the lipid portion is going to be in towards the center. We'll talk about, you know, what can get in and out of there without permission. It's got to be non-polar, no charge. It's got to be, if it's lipid soluble, it can. And if it's really, really tiny, it can go in and out without a bodyguard or these integral proteins allowing it in and out. All right. The cytoplasm is the intracellular fluid containing the organelles. So cytoplasm, usually cyto, when you see that, it's going to sort of mean like inside of something, a fluid inside of something. All right, so this is kind of aqueous solution that all these organelles are small organs or components of the cell. They live in there. All right, and the nucleus, I'm hoping you've all at least heard of this, is a DNA containing control center of the cell, and this is what's going to control reproduction of the cell, or what the cell does or doesn't do. And we're going to find out when things dock on the cell, if them to do things, it's going to be sending signals to the nucleus. The nucleus has all that genetic information. It'll start replicating that information and sending RNA out of the nucleus into the cytoplasm for construction of whatever that protein or enzyme or lipid or whatever it happens to be, depending on which organelle it's attaching to, and, you know, translation, transduction, and we'll talk about all those different components of that. All right, so here's the structure generalization of a cell. So this is in pretty much every textbook, from human bio to microbiology, or even pathophysiology. So it's all a general idea or general structure. We have models of this here at school. And please realize right now that this is pretty much every organelle or every part of a cell, but realize not every cell has these things. Or, you know, here's Golgi apparatus, just as an example. So if a cell was going to produce a lot of things and it had to ship it out through exocytosis, it's going to have a lot more Golgi apparatus. Here's some rough endoplasmic reticulum, which is studied with ribosomes, and these are what where you make your proteins. The ribosomes are what take the nucleic acid, or the nucleotides and nucleic acids, and we put them all together and make the protein. So if you're cell is going to make a lot of proteins or enzymes or whatever. They have a lot more of these. All right, here's a mitochondria. Yeah, from high school, you probably learned this is a powerhouse of the cell. So this is what's going to actually take oxygen and glucose, basically, and make adenosine triphosphate to produce energy for the cell. So if you have a cell that is constantly moving or needs a tremendous amount of energy constantly, it's going to have more mitochondria. All right, and mitochondria can, for the most part, they can regenerate on their own. So if a cell decides it needs a lot more energy, it can automatically make more mitochondria. It doesn't need permission, doesn't need a signal from the nucleus. It can do that. Nucleolus, and we'll talk about this with mitosis, how chromatids and how these things kind of coil and uncoil with the genetic information, the DNA, for reproduction of the cell. Smooth endoplasmic reticulum, these are going to be usually for lipids or fat breakdown or detoxification or cleaning. So if it's a liver cell and it was doing a lot of cleaning or whatever, or if it's something needed to produce a lot of lipids, it would have more of those. All right. Centrioles, all right, these are going to be used for regeneration of the cells, and you know, as they get smaller in number, the cell will just be unable to replicate. You know, as these get smaller and smaller, it determines whether they can replicate or not. There's a peroxisome. All right, so this has peroxide, or it has some enzymatic peroxide. So oxygen's toxic to some of these organisms. So peroxisomes, they kind of, if the cell was a macrophage and engulfed it, it would hit it with these peroxisomes. It would digest whatever it happened to be. Or if it was a bacteria, it was some kind of a neutrophil or some kind of white blood cell, it would engulf the bacteria. It would hit it with the peroxisome. It would literally start digesting it, destroying it, and then the cell would start picking up the components and start reusing them. But that's beyond the scope of the course. Here's secretory vesicles. All right, so these come from the Golgi apparatus. They literally dock to the biphospholipid layer, and they merge with it, and they open up and they excrete whatever the substance is. Now, it could be cellular debris, or it could be some kind of substance that cell produce or need it. So it would push into the extracellular fluid, and then somehow it'll get back into the capillary bed and be distributed, shipped out to whatever part of the body, right? So for this, you know, this portion of the chapter, I really need you guys to just learn all these different, hopefully it's review, learn all these different organelles and kind of what they do, all right? So that's kind of important to know. All right. So extracellular, I learned last time, this is extracellular. So extra, so it's outside of the cell, right? So we realize that all of your cells are floating around in this fluid. And this fluid generally is a constituent of your capillary beds and flowing of your blood. So it's this fluid it floats around in. And this fluid should have the nutrients supplied by your capillary bed, and it also has some of the waste products from cellular metabolism that has to go back into the blood, and we recirculate that. And I said before in chapter 1, I think, that your liver and kidneys are going to be filtering that out, right, if they're functioning properly. So substances found outside of cells, extracellular, outside cell. Classes of extracellular materials include extracellular fluids, so this body fluid. So anything that's not literally inside of a cell is considered extracellular, outside of a cell. So if it's not part of the cytoplasm, it's extracellular. We're also going to talk about interstitial fluid. That's fluid the cells are floating around. But extracellular fluid can be anything that's not in a cell. So it could be fluid in your capillary bed. It could be fluid in your synovial joints, the bursa sacs in your between your muscles and tendons for lubrication. So any fluid that's not literally inside of a cell is considered extracellular. All right, so just kind of get that into your head, and I'll be repeating it over and over again. So interstitial is inside of parts of your body that your cells are floating around. And extracellular is just anything that's fluid that's not inside the cell. And when we start talking about dehydration and fluid balance, just realize if you're dehydrated or you're losing fluid, your body will actually start pulling it from your cells if it has to. Right? So interstitial fluid, cells are submerged or bathed in this fluid. Blood plasma is fluid of the blood, and cerebral spinal fluid, all right, fluid surrounding your nervous system organs. So that's in your central nervous system and spinal cord, all right? When we really start talking towards the end of this course, we're talking about nervous system and special senses. Please realize that your hypothalamus or areas of your brain that are controlling your body functions or regulating homeostasis, they're going to be looking at the cerebral spinal fluid, and they can tell by the constituents or the components of that cerebral spinal fluid what your body has a lack of or superfluous or extra amount of. And it can kind of tell whether you're dehydrated, or it can tell the glucose level, it can tell hormone levels. It'll decide to turn things on or off to keep that homeostasis or that equilibrium balance. So it's really amazing that your body can do this. You don't even have to think about this. All right, so cellular secretions, saliva, mucus, those are all contain fluid. Your goblet cells are going to produce mucin, all right, and then when they come in contact, mucin, that protein constituents, when it comes in contact with water, it turns into this mucousy or slimy kind of jello for lubrication, and also is kind of like flypaper. It causes things to stick to it, and then they can be engulfed. Your cilia kind of moving around, and then hopefully, you'll swallow it, and your stomach acid will kill it, or your macrophages will go in through phagocytosis. They'll go in and consume it and destroy it. Right? Your extracellular matrix is substances that act as glue and hold your cells together. So these are extracellular, there's fluid all around your cells, and they kind of, proteinaceous, or it's sort of like semi-solid clear jello. I mean, you guys may know what jello shots are or whatever. But until they solidify, they're very, very thick. And it's going to hold these cells together, all right? Kind of a very, very thick gelatinous type material, all right? So very, very key component is your plasma membrane. It acts as an active barrier separating the intercellular fluid from the extracellular fluid. So we'll see a picture coming up. Inside the fluid, the cytoplasm is intracellular. Anything outside the cell is extracellular. So when you're reading, or you're taking a quiz or a test or a homework or board exams or MCATs or whatever it is, so just start thinking, what are they asking me here? Plays dynamic role in cellular activity by controlling what enters and leaves the cell. So extremely important. Your plasma membrane determines what is allowed to come into your cell and what leaves your cell. And remember, this is a biphospholipid layer, and it is non-polar. All right, so that means it has no charge. So you guys know the North Pole, South Pole, they have certain charges. All right, non-polar means it has zero charge. All right, so if it had a charge, positive or negative, it would attract, if it had a positive charge, it would attract things with a negative charge. And if it had a negative charge, it would attract things with a positive charge. It has zero charge. It's electronegatively neutral, all right, and it's biphospholipid. So once again, if something has a negative charge, it can more or less go in and out. If it's very, very tiny, it can go in and out. And if it's lipid soluble, it can go out of cell. So those are three things generally that can go back and forth through this plasma membrane without specific carrier proteins, or without specific bodyguards, or certain things, carrier molecules like insulin can get things in and out of the cell. All right. Plays dynamic role in cellular activity by controlling what enters and what leaves the cell, also known as a cell membrane. So it's going to be interchangeable. So whether we're talking about us as eukaryotic cells or microorganisms as prokaryotic cells, they all really still have a cell membrane. So that's pretty common in this, well, this universe, I guess we could say. Right? So the structure of the plasma membrane consists of membrane lipids that form a lipid bilayer. So from now on, when you see lipids, all right, from your chemistry package or whatever, you should realize these are fat lipids, all right? And lipids really don't like to dissolve in water. They'll float on top of water. There's a different density. So if you don't believe me, go home and get some Italian salad dressing in your refrigerator and look, it'll be separated. So before you use it, you're going to shake it up and emulsify it, get it emulsified temporarily and put it in there, all right? But we'll talk about lipids later, and we'll talk about digestion and things. But for right now, just realize the middle layer of this biphospholipid layer, and we'll see a diagram, is going to be the lipid component. Specialized membrane proteins float through this fluid membrane, resulting in consistently changing patterns. It's referred to as a fluid mosaic pattern, so mosaic tiles or whatever. And you'll see this in diagrams or whatever. Please realize these can float around and they can move. All right, these proteins, they bob up and down and they can move. Yeah, we'll get to that later. Okay. Surface sugars are glycocalyx, depending on how you pronounce it. Glycogen, glucose, glyco. Always when you see that, it's going to be carbon, oxygen, hydrogen. So just think of it as some kind of a sugar or CHO, carbon, hydrogen, and oxygen. It's going to be some component of that. Usually it's a 1-to-1 ratio. So 1 carbon, or 2 hydrogen, 1 oxygen. So it's going to be 1-to-1 ratio. All right. Membrane structures help hold cells together through cell junctions. Right, so these membranes will come together. Fix this light. Right. They're going to come together and they're going to attach. Right. So we want these cells to be able to attach, and especially if they're muscle cells or they're under a tremendous amount of force or peristalsis, like in the digestive system. As these move, we don't want them to tear apart. All right. So we're going to talk about gap junctions, tight junctions, desmosomes, how they can rivet these things together and hold them together so fluid can't leak in between the cells. And when some of these myocardium or cardiac muscle cells contract, they're not tearing them apart, they're really holding them tight together. Okay. So here's your biphospholipid membrane. I think I can do control plus, yeah, nothing. Plus 0. Okay. All right, we'll deal with it. Okay. All right, so the plasma membrane is a phospho, so it's phosphate. Lipid is that fat, all right, layer with embedded proteins arranged in a fluid mosaic. So these will literally float around, all right? So this is your phosphate, and your lipids, biphospholipid. All right, so just kind of, I know all these things over and over again. Lipid in the middle, phosphate in the outside. So phosphates, they are water loving. All right, so that's why this is extracellular, intracellular. This is fluid, so they're water loving. So they're going to line up. They like the water. So they're going to, no matter what you do, they're going to line up with the phosphate end towards the outside or the inside where the fluid is, and the the water fearful part is going to stay in the middle. The lipids are always going to stay in the middle. And these are your proteins that are floating around. These float around, they can bounce and move all around. All right, but you're going to look through here. You see kind of a channel here. This is going to be able to bring things into the cell. I said before, if they're negative or lipid based, they can get in and out, or small. All right. So if we had something really, really large, and maybe it had a positive or negative charge. It was a really big molecule. If we needed, through diffusion, if we needed in there, depending on the concentration gradient, we wanted to bring it in there, this will allow it to come in. Sometimes we can bring things in, and as they come in, we actually can change their chemical or formula, and we bring them in as a whole new molecule. This kind of saves time rather than doing it inside. We automatically know we have to attach another molecule to this. As we bring it in, we just change it. It's just it's how incredible the human body is. We can do that. Right? Right, so I can't, sorry guys, I can't get this thing to magnify on my MacBook. So I can't blow it up for you. I can't just zoom it like with my phone or whatever. So we'll have to deal with it. All right. So these videos, when you go to reduce, these should play. They're dated, okay? So they're literally 20 years old, just about, but the information is still the same. And they're short and they're relatively accurate. So start visualizing this. All right. I'll just play one for you. So all you have to do is click on that button there.
The plasma membrane is selectively permeable. It lets some substances through and not others. The hydrophobic interior of the bilayer is one reason membranes are selectively permeable. Hydrophobic molecules, those that are soluble in lipids, can easily pass through the membrane. In addition, small molecules like O2 can sneak between the phospholipids of the membrane. On the other hand, large hydrophilic molecules like glucose and ions, such as sodium ions and hydrogen ions, cannot pass through the membrane unaided.
So they can't get through. So they're going to need some kind of, well, glucose, obviously, everybody should probably know, needs insulin. These are carrier proteins. And then, before I forget, guys, what you were told before, insulin is not just for glucose. It can bring a lot of things into a cell. It can actually carry it in, even pharmaceutical agents or some chemotherapy agents. If you attach it to insulin, it can actually bring it into the cell. It'll dock on certain specific markers on cells that may just be on your cancer cells, and we can selectively pick just cells that have their surface proteins altered due to cancer. We can bring it into those cells specifically. Right? So the membrane lipids, so when we say membrane lipids, we're thinking biphospholipid layer and the lipids, the fat portion of that, or the lipid portion. The lipid bilayer is made up of 75% phospholipids. Those are on the outside, which can consist of two parts. So remember the popsicle head or whatever is going to face the outside or inside. It's water loving. All right. Hydrophilic, love, phil means love. It's on the outside, so it's going to line itself up towards the outside or inside. Phospholipids are polar charged, so they're hydrophilic, water loving. Fatty acid tails are non-polar, no charge, so they're hydrophobic, water hating. All right. So, all right, the phospholipids are polar or charged, so they're hydrophilic, water loving. That's why it loves water, because water is has a large negative. All right, so oxygen is a huge molecule. The two hydrogens are tiny, so it's going to have, the charges are going to be not distributed quite evenly. All right, so it's polar or charged. Fatty acids are non-polar, and that's why certain things with no charge can actually sneak through that. The glycolipids, so glyco, remember sugar, lipids, so fatty sugars, lipids with sugar groups on the outer membrane surface. All right, sometimes these can be antigens or markers, or sometimes they can be attachment sites for certain enzymes to signal the cell to produce or not produce something. All right, and cholesterol, everyone says, oh, you know, cholesterol is terrible, it'll kill you, whatever. You need cholesterol. Increases membrane stability. It's actually a structural component for all of your cells. Right? And I mentioned before, we need this also, lipids and cholesterol for your biphospholipid layer. And we also need some of that for the myelin sheath and some of your neurological development. We need some kind of structure. All right. And then even you know, other organisms of estradiol, anything that ends in OL, they have these structural proteins in their biphospholipid. Even microbes have it. They need some kind of structure. Ours just happens to be cholesterol as well, what our body uses. So even if you're taking these cholesterol medications that block synthesis in the liver or whatever, or you're stopping consumption of it, your body is going to do whatever it can to produce them because it needs these cholesterols. If you are going to go into any kind of cellular repair or as your cells regenerate, they have to have cholesterol in them. Right? So the structure of your plasma membrane, the polar or hydrophilic, includes a phosphate group. So remember, those are always going to line up on the outside. All right, just visualize it, picture it, know why. It's going to be the central dogma of biology and how your cells operate. Right? So membrane proteins. So, you know, why would these membranes have proteins in them? Well, proteins are going to be very large structures. They're going to be able to transport things in or out, and they're going to be like gates or security guards to allow things in and out that we want. Right? Allow cellular communication with the environment. So these membrane proteins can tell the outside of the cell what's in the surroundings. So whether we know if it's, you know, we won't get into the detail, but if it's a viral component and we have the interferon, it's going to signal the cell to say, you know what, listen, if this virus gets inside of you, I want you to go through something called apoptosis. I want you to go in and kill yourself so that the virus can't get inside the cell and replicate. Right, just an example, we'll get into that later. All right, make half the mass of the plasma membrane. All right, so they're there. And remember, these integral proteins can float around. They can move if they need to. And here's the thing, if we need more of these membrane proteins, the body can just make more. If we need more receptors, let's say we need more receptors for insulin. Somebody is a non-compliant diabetic and they keep consuming all this sugar and it's floating around in the blood, and when, you know, it's not good to increase the osmolality of your blood, but if we can't get the glucose into the cell, your cell will just make more integral proteins, make more receptors for the insulin to dock on and bring more sugar in. But at some point in time, you can't fit anymore, so then you have a major problem, and we have a problem with that with non-compliant diabetics. So most have specialized membrane functions, so they're all going to eat redocking stations, HLA markers, or HLA, or that's good enough for now. So markers that are very specific to you. Right. Some float freely and some are tethered to intracellular surfaces. Right. Some of them bounce around freely. Some of them are tethered by these integral collagen fibers. They're kind of like structural proteins. Like some of these molecules can zip line on them, and we'll see pictures of that coming up. All right. So integral proteins and peripheral proteins. Integral generally are docked in and they're non-moving. They're for structural integrity. Peripheral proteins are just floating around and they can change shape for the most part. All right, integral proteins are firmly inserted into the membrane. Most are transmembrane proteins. So trans means they go from the extracellular fluid all the way through into the intracellular fluid. All right, so that means if there's a hole or a lumen or a tube, we can bring things through there. All right, or sometimes they're doors. So once something attaches to them, they engulf it, open it, and they open up. So they don't allow things with certain size or shape to dock, and then they'll allow them in. All right, if it's like an aquaporin, these are open channels for fluid or water to go back and forth. Or if we're talking about the sodium-potassium pump, we have very, very certain integral proteins that will allow theoretically 3 sodiums out, 2 potassiums in. So we can keep running that chemical gradient. All right, we always want the inside of the cell to be slightly electronegative as opposed to the outside of the cell. Right? And I will get into that later. So I apologize, guys. I'm just kind of going ahead, but I want you to start hearing these things over and over again so that when we get to these things, you know, maybe you've heard it before, or you'll have a reason of why we're learning this, or it's for the future. We just kind of learn these concepts now, why we're learning it, and we'll relearn them when we have to relearn the nuances of that, all right? All right, so they have both hydrophobic, remember phobic is sphere, hydro, you know, think of fluid, all right, hydro, you know, unless you're Canadian, and then hydro means electric, or national grid or whatever in this area, all right? So they have a so a water fearing and a hydrophilic water loving region. Remember, hydro, it's fearing water, so it's going to be in the middle of that biphospholipid layer. Hydrophilic, if it loves the water, is going to be on the external or the internal surface. Functions as transport proteins, channels and carriers for enzymes and receptors. All right, we'll learn about enzymes, and I'm sure you've heard about them. So enzymes are things that can help chemical reactions run faster. They kind of lower the activation energy of certain things. Right? So peripheral proteins, remember I said these are on the periphery, so they're not really attached. They're kind of not tethered. Right? So if you want to think of them as like a bobber, anyone's gone fishing, not really like buoys in a channel for on the river or in a lake. Like, so those are literally tethered to the bottom of the plane of water. All right. So loosely attached to integral proteins include filaments on the intracellular surface used for plasma membrane support. So these filaments are attached inside the cell, anchored down there. So there's going to be support. And remember, you know, we kind of think of cells theoretically as these round spheres, right? But they're really not. Each cell sort of has a certain shape to it, but we want it to have a certain shape. So we want these, you think of them as tent poles, or we want to have some kind of structure. It's got to be a little bit pliable. We have to have some kind of structure in these cells. So these function as enzymes, motor proteins for shape changing during cell division and muscle contraction. And we'll get into that when we start talking about mitosis, meiosis, really more towards the end of ANP2, and muscle contraction. We'll talk about cross bridge and sarcolemma and cross bridge, and how muscles can contract, and they kind of have this rowing or ratchet motion where they contract and they shorten, causing muscle contraction. All right, and we want to have cell-to-cell connections. So sometimes we want these cells to communicate back and forth, and especially if they're, let's say they're a myocardium and they all have their own conduction system. We want these cells through specific gap junctions called intercalated discs. We want them to be able to tell the cell next to it contract, 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 kind of like doing the wave, if you've seen it at a sports game, or here in Orchard Park, we're right near the Bill Stadium. So, you know, it's common knowledge over here. All right. All right. All right, so these are transport proteins. So if you look here, remember I said these are going to be channels that are completely open. So these are kind of going to cause things to float back and forth, contingent upon their concentration gradient, whether it's active transport or simple diffusion, or you know, let's say it's a kidney cell and it's running against the concentration gradient. So we're talking about some of those specifics. All right. So protein on the left here. Let me see. Yeah. Oh, good Lord. Okay. What have I done? Yeah. All right. So on the left here. So these are. All right, I guess that's not going to work. I need more practice. All right. So here, that spans the membrane, may provide hydrophilic channels across the membrane that are selected for a particular solute. All right, so solutes are things that are dissolved in a solution. So if it was sodium, all right, that would be the solute. If it was potassium, it would be dissolved in that extracellular intercellular fluid, and it would move back and forth, all right? Some transport proteins on the right hydrolyze ATP as an energy source and actually pump substances across the membrane. Hydrolysis is when you cut or cleave things using water. We'll get into that, all right? But for ATP, you know, I kind of alluded that we're going to send sodium out, potassium in. So theoretically, you always want to picture 3 sodiums, Na plus on the outside, and then 2 K pluses on the inside. And if you can think about it, there's 3 pluses on the outside, 2 pluses on the inside. So the outside of the cell is going to actually have a more positive charge, or the inside is going to have a slightly negative charge as opposed to the 3 outside. So it's going to be slightly less or slightly negative. All right. Not a big deal for right now, but at least you've heard it. All right. So these open and close contingent upon whether we want them to. We don't want these things free floating. We always sort of want to have a slightly positive charge on the outside, slightly positive charge on the inside. So when we want that cell to do something, we actually switch the gate, the positive come in, the negative go out, and we literally, like flicking a switch on and off, we can actually change the polarity of that cell membrane and cause things to happen. You guys can watch these. It'll actually go in and show you. And like I said, they're 20 years old, but the animation's not bad, and it's going to give you a very simplistic example of what they do. So start visualizing this stuff. Okay. And then we have receptors for signal transduction. I said, you know, so these are going to have a bunch of signals, right? So a membrane protein exposed to the outside, extracellular, may have a binding site that fits a shape of a specific chemical messenger, such as a hormone, right? Hormone or enzymes. And remember, these are going to have a specific shape. So let's say your endocrine system pumped out a specific hormone into your bloodstream, all right? The cool thing is that hormone knows exactly what cell it has a specific marker for. It's kind of like your key fob. It knows, well, it doesn't know unless you show it, but it knows what lock and key mechanism fits. So these chemical messengers will literally float around your bloodstream until they come to the area of the body where these cells are located. They come out of your capillary bed into that interstitial fluid, attach to the cell, and turn that cell on or off or cause it to do something. All right, so you guys may think, well, why didn't you know that? Well, think of how amazing that is, that without even thinking about it, your endocrine system can make a hormone that can float around your blood. It actually knows the chemical gradient, where to leave your bloodstream, attach to your cell, and turn that cell on to make a specific protein or another hormone. So it's completely amazing. And these are protein receptors that are just floating on that cell, and they know when this chemical messenger attaches, they open up or they send a signal to the nucleus, and the nucleus makes whatever it makes. It starts replicating your DNA into RNA, goes through introns and extrons and starts cutting and cleaving a very specific portion of that DNA to send it out to the organelles in your cell to produce something very, very specific. So absolutely fascinating. All right. When bound, the chemical messenger may cause a change in shape in the protein that initiates a chain of chemical reactions in the cell. And so we can have first messenger, we can get into second messenger and cyclic AMP and all that other stuff that you guys just don't need to know about. You're going to hear about it, so you kind of know. First messenger just means it attaches, it does something immediately. Sometimes it has to have a second messenger. So it changes something here. By the time it gets to here, it turns into something else. That's second messenger, all right? And then the second messenger goes to the nucleus and does it. All right, so if you ever hear that, that's kind of what it means, simplistically, all right? And enzymatic activity. A membrane protein may be an enzyme with its active site exposed to substances in the adjacent solution. All right, so it could be the extracellular solution or it could be an intracellular solution, right? A team of several enzymes in the membrane may catalyze sequential steps of the metabolic pathway as indicated, all right? So we can have, you know, A plus B equals C, or B and A, you know, we can combine these two together depending on the enzymatic pathway, right? We can change one thing to another, or we can get a substrate and a receptor, and as they come together with coenzymes, they can reshape something, and these proteins will cause these things to link and attach and then change. And it only happens when needed. It has to have chemical signals through that homeostasis or that negative feedback loop to bring you back to homeostasis. So it's really, really pretty fascinating what your body can do. All right, and then we want cell-to-cell recognition. Some glycoproteins, proteins bonded to short chains of sugar, which help make the glycocalyx. Remember I said glyco is sugar, proteins, serve as identification tags that are specifically recognized by other cells. You know, so simplistically, you could say that these cells want to know that they're related or that they're normal or whatever. So we could theoretically say, if we had some of these membrane proteins that were altered or didn't seem quite right, your immune system, if it was working, would send in NK natural killer cells, or it could send in activated memory T cells. If it sees that marker, goes and destroys it. You know, we won't get into a huge amount of immunology at the moment, but please realize that it's also another key component, especially if you go into healthcare. You have to know how your body, the immune system functions in your body. It's extremely important, because without it, well, then you have illness and sickness and disease and you know, all kinds of things. All right, so there's some of these are attachments to cytoskeleton and extracellular matrix. All right, so you'll see here we got some protein. There, this is the extracellular matrix. They're attached there. All right. Some of these are attached to the internal cytoskeleton. So this helps maintain the shape of things. So it's a little bit pliable, but it has some structure. And then later on, we'll see that some of these things inside your cell can literally, they can zip line across this. You know, the shortest point between two things is a straight line. So they can actually zip line. They don't have to swim around. They can just grab this and zip line right to wherever they want to go. So fascinating. All right, and then cell-to-cell joining. All right, so membrane proteins of attached cells may be hooked together in various kinds of intracellular junctions. All right, so if you look, this is literally, you got to visualize this. This is intracellular biphospholipid layer of a different cell, completely intracellular. So these two cells have attached to each other. All right, and we may want these to communicate with each other. So if it's a myocardium or intercalated discs, we want to send a signal propagation from one cell to the other, so it knows what to do in a certain sequence. So your heart will beat consistently and accurately. Or we may want these two cells to adhere together very, very tightly. So if they were storage or excretion or part of if they're part of like the nephron of the kidney, or if they were part of part of another structure that was moving, or if it was in the digestive tract, we wouldn't want fluid components from the digestive tract to get in between these cells and separate it. We wouldn't want digestive enzymes to leak in through here and start separating the cells, because now you'd have Crohn's disease or IBS, or you'd have some kind of disease where you're destroying your microvilli or altering the surface area of your GI tract, causing all kinds of problems. So just kind of realize we also can have these proteins connecting these cells together, either adhering them tightly if they're under a lot of pressure or movement, or we want them to communicate with each other so they know what's going on. All right, because they can't, there's no Wi-Fi or internet or pagers or any of that stuff. They have to be able to communicate through chemical messengers. Glycocalyx. So glyco is always sugar. So kind of think this as a candy coating, if you want to think of it as candy coating an M&M, you know, whatever you need to do to visualize this, that works, right? So it consists of sugar sticking out of the cell surface. Some sugars are attached to lipids, those are fats, and some to protein glycoproteins. Okay? Every cell type has a different pattern of the sugar coating, right? Functions as specific biological markers for cell-to-cell recognition, allows the immune system to recognize self and non-self. So there's glycocalyx for these sugars and proteins, and we have HLA, human glycolytic antigens, or MHC1 markers, major histocompatibility complex. Same exact thing. These are very, very specific markers on your cells that tell your immune system that you are self, all right? So if these ever alter or get changed through cancer or any other deleterious thing, your immune system will say that marker doesn't look right. It'll go in and super tag it, or it'll start grabbing part of it off, the antigen, and start presenting it to your immune system and say, listen, I found this, it's foreign, find me an antibody to it, get me some memory cells to it, let's notify our CD8 cells, and go in and destroy that too, all right? And these are also the proteins, if you had an organ transplant or a bone marrow transplant, if you transplant an organ or whatever, and these glycocalyx markers, biological markers, are different, all right, your immune system is going to go in and attack that. So you give them immune suppressive drugs. All right, so if you were going to go in and do a bone marrow transplant or any kind of organ transplant, you want to find an organ that has these cell markers that are as close to yours as possible. And I think there's like 9 different, I'm probably wrong. There's like 9 different components of it. So if you have 7 out of the 9, it's a good match. Or, you know, sometimes people will say, you know, I was a kidney or organ donor and the match was completely perfect. Well, it may not be completely perfect unless you have an identical twin, but it can be so close that they wouldn't anticipate any problems, or wouldn't anticipate your immune system not recognize these glycocalyx markers as your own. Right. So please realize too, before we get into genetics, we're all made up of the same exact proteins and amino acids. All right? So all the codons are they're the same. The sequence is the same for these certain proteins, but please realize we all are completely different and unique, and our epitopes and our DNA are all completely different. Right? So glycocalyx of some cancer cells can change so rapidly that the immune system cannot recognize the cell as being damaged. All right, so mutated cell is not destroyed by immune system, so it's able to replicate. So what they're saying here is, if we go in, cancer cells go in, and the cell is altered, the DNA is altered, the sequences are altered, it can start changing the markers on the outside of the cell, those glycocalyx markers, and that's good because your immune system's going to say, okay, you don't look quite right, we're going to go in and destroy you before that cell can keep reproducing. All right. The other issue is if that cell goes in and has all these changes and it doesn't change the markers significantly, the immune system cell looks pretty normal to me, it leaves it alone. Now that cell is able to reproduce, go through mitotic division over and over and over again. Cancer cells don't have any contact inhibition, or they will just start going through mitosis for no apparent reason, just over and over again, whether they're needed or not. All right. So they just go replicate insane. Right. So cell junctions. So these are the things that are between these cells, and realize there's different ones for different reasons. All right. So some cells are free, not bound to others. So blood cells, your white blood cells, red blood cells, platelets, natural killer cells, these cells are floating around either in the tissue, whether they're antigen-presenting cells in your tissue or mast cells in your tissue. They're floating around for a very specific reason, or sperm cells float around. All right, every other cell generally, for the most part, is attached to other cells, and 2 different cells type make a tissue, 2 different tissues can make an organ. So on a cellular level, we want these cells to stick to each other and have a purpose and have a commitment to each other for integrity and to stay in space, all right? So most cells are bound together to form tissues and organs. Three ways cells can be bound to each other are tight junctions, desmosomes. So tight junctions are things that are very, very tight. They don't want to let fluid in or anything in between these cells. Desmosomes generally are like rivets. So these things hold, they're like, rivets, or rivets, like nuts and bolts or rivets that hold things together. So anything that's under a tremendous amount of torque or tension, we don't want these cells separating, all right? And gap junctions are what I showed you before, kind of, they're gonna be gaps or holes, communication channels between cells. And we want that there so we can propagate a signal, or these cells can communicate with each other for a specific reason, right? So tight junctions are integral proteins on adjacent cells fused to form an impermeable junction that encircles the whole cell, right? Prevents fluid in most molecules from moving in between cells. We want things super tight. We don't want things getting in there, you know, I don't want to say delaminating, but causing some kind of a change, right? Where might these be useful in the body? So generally, on exams or whatever, they're gonna give you some kind of a scenario where these cells are gonna be surrounded by some kind of extracellular fluid that we don't want getting in between the cells. So it could be GI tract, could be bladder, could be kidney, or somewhere in there where we don't want the fluid surrounding it to wedge its way around between the cells, all right? All right, so this is kind of showing you, like this is the GI tract. So this is your microvilli for surface area. We don't want the digestive enzymes and things to get in between the cells. Right? So we want these tight junctions. All right, they're kind of like Ziploc bag liners all the way up and down the cell. All right, desmosomes. So these are the rivets. All right, rivet-like cell junctions form when linker cat adherens, adhesion, neighboring cells interlock like teeth of a zipper, right? Linker protein is anchored to the cell through thickened button-like areas on the inside of the plasma and as plaques, right? So, you know, you can have plaques on the wall, you can have plaques on your teeth, but now we can have plaques inside the plasma membrane. All right, so these are, you know, kind of like if you, you sew a button on something, you have the button and then you have a thing on the back that you anchor it to. So just think of it as something structural that you can attach it to. And plaques, if you have plaques in your arteries or plaques on your heart valves or whatever, these are dense materials, all right? Keratin, right? So keratin is a protein that's a component of your skin, your cells. Filaments connect plaques intracellularly for added anchoring strength. All right, so just think of that as additional protein or some kind of substance that we can anchor to. Desmosomes allow give between cells, reducing the possibility of tear under tension. So these are rivets, and if these are under contraction, the cell can be flexible, all right, but it's not going to pull apart. Where these might be useful, so think of anything that had strong contractile forces. So myocardium or heart muscle. You know, you could think of any kind of a sphincter, pyloric sphincter in your stomach, or anal sphincter for defecation. Anytime when there's a lot of pressure, we wouldn't want these cells to rip apart. All right. All right, so gap junctions. So these are transmembranes. So they go between membranes, and this is between two different cells. We want these cells to be able to connect and communicate with each other. Form tunnels that allow small molecules to pass from cell to cell, used to spread ions, simple sugars, and other small molecules between cells. So spreading ions. So these could be ions or anything with a charge, positive or negative. We want small ions to pass. So we can, by changing these ions, we can send calcium signals, sodium signals. We can change the polarity, electrical charge, we can cause things to depolarize or propagate a signal doing this, right? Allows electrical signals to pass quickly from one cell to the next using cardiac and smooth muscle cells. So cardiac muscles, we're gonna learn, your heart has its own pacemaker. I'm sure you've heard of this. So it doesn't need the brain to tell it. It automatically knows when to propagate that signal, and each cell fires when it's supposed to. So we have complete contraction of the aorta, then the ventricles in a very, very specific order, right? And smooth muscle cells, those are for sphincters or digestive tract. We want your digestive tract to, we have longitudinal and circumferential fibers. We want that to contract. All right, squeeze, and then it blocks it, so it can't go backwards. We squeeze it, and we block it, squeeze it, and then block it, so that we have peristalsis. We have food moving from your stomach to duodenum to your jejunum to your large intestine, so that things don't back up. We want that signal to propagate in one direction. All right, so if you had retroperistalsis, that's when you have a blockage and everything reverses, and then you start vomiting up through your stomach and esophagus. And you can imagine what that's got to be like. Right. So it's just kind of showing you that these are connections between cells so they can communicate with each other. Right. So structure of plasma membrane. So 3 of 3. So this is the third portion of it. Many substances must constantly move across the plasma membrane. Some molecules pass through easily, some do not. So plasma membrane is selectively permeable only to certain molecules. And remember, I said the plasma membrane is selectively permeable too. Very small, lipid, zero charge, very selective. Otherwise, it will allow some things in, but only under certain conditions. If we need it, it'll come through. We don't want it to come through if it's going to be deleterious or a problem, or if the concentration gradient is already saturated, right? So 2 essential ways substances cross the plasma membrane. We have passive transport. So passively, you know, people are very, very passive. There's no action required. No energy is required. It's really passive. It just happens organically through concentration gradients. It's kind of what we want. Active transport, though, if we have to be active about it, we need energy. ATP is required. Adenosine triphosphate. So active transport is usually when we have to bring across very large molecules that are going to, you know, need energy, like we have to ship it across, or if we're going against the concentration gradient. So we're going against the laws of nature, and that's it happens in the kidneys and some, the distal proximal, convoluted tubules, proximal. Yeah, so those areas of the kidney, we can fine tune things, but sometimes we have to move things across the concentration gradient, and we have active transport. Or if we're moving very, very large molecules, we need some ATP. So that's called active transport. All right, passive transport requires no energy input. 3 types: simple diffusion, facilitated diffusion, and osmosis. So I'm hoping you've heard of simple diffusion or osmosis. And simple diffusion, if you don't know what that is, it's when you have something concentrated, you leave it in its natural environment, it will spread out until everything is completely spread out completely evenly. So think about, you know, if you were to, you know, spray cologne or something in the room, it's very dense where you sprayed it, but over time, those molecules will disperse to every corner of the room. When it's all completely equally distributed, it'll stay in that concentration, all right? Or if you were to put, classic example, you put a sugar cube in a beaker of water, it's going to start dissolving, and then at some point in time, if you leave it, that glucose solution, or sucrose, would be distributed completely evenly. So the concentration percentage of sucrose in the solution would be even no matter where you took a sample of that water. All right, all types involve diffusion. natural movement of molecules from areas of high concentration to areas of low concentration, right? So natural movement of molecules inside the solution from areas of high concentration to low concentration. So remind yourself, if what's easier for you to go from upstairs to downstairs or from downstairs to upstairs. So yeah, it's always easier to go from upstairs to downstairs. You're going from high to low, it's always easier. So just remember, you know, whether when you're talking about diffusion or any of these things, remember it's going to be natural movement of molecules from areas of high concentration to low concentration. So if you have a solution that's 1% sodium, or a solution that's 10% sodium, right? 10% sodium is higher molecules. So there's more molecules in it, there's actually less water, right? So if it's 10% sodium, 90% of it's water. If it's 1% sodium solution, all right, it's 99% water. So just keep that in your head. Also referred as moving down the concentration gradient. So things are always natural movement of molecules from areas of high concentration to areas of low concentration. So if you put those two solutions together, a 1% solution of sodium and a 10% sodium solution, and you had a diffusing membrane, what's going to happen is the 10% solution is going to move into the 1% solution until you get to a concentration of about 4 point, I'll make this up, 4.7, all right? Because half of those 10% is going to go into the 1% until the concentration of sodium is equal on both sides. So all molecules have random high-speed movement due to their intrinsic kinetic energy. So all molecules, whether it is cologne or sugar or whatever, they're going to move until they bounce across a barrier until they're all equally distributed, and they just stop. All right, movement results in collisions between molecules. Molecules in higher concentration areas can collide more, resulting in molecules being scattered to lower concentration areas. So remember, if something's in a higher concentration than more molecules, it's moving, it's hitting like billiard balls or whatever, it's bouncing, it's gonna keep moving them to areas where there's less molecules because there's nothing blocking it. At some point in time, they're gonna be bouncing all over until the concentration of the molecules is equal everywhere, and then there's no motion because everything is equidistant from each other. Right. So here's diffusion. Like let's say I put a dye pellet or a sugar cube or whatever, put it in there over time, it's going to completely diffuse until the concentration is completely equal. All right, so passive membrane transport. Speed of diffusion influenced by 3 factors. So passive membrane, so it doesn't take any energy, so there's going to be some factors. Concentration, the greater the difference in concentration between the two areas, the faster the diffusion occurs, right? So if you have one solution that's 1% and one solution that's 2%, it's going to take a long time. But if you have something that's 1% and something that's 50%, the diffusion is going to happen a lot faster because the concentration between the two is great. The span is much, much greater. Molecule size. Obviously, smaller molecules diffuse faster, they take less energy, they move quicker. All right. And then temperature. Higher temperatures increase kinetic energy and result in faster diffusion. All right. So just like your body or even us as humans. So as the temperature gets warmer, we have more energy or we want to move more. All right. When it's colder, we don't feel like moving. The only time we're going to move faster is because we want to produce heat, right? All right, same thing with your body. As your temperature increases, your metabolic rate goes up, your immune system function goes up. But as you heat things up, things dissolve faster, right? Okay, you know, you want to think of Jell-O, all right? So Jell-O, it doesn't dissolve very quickly in cold water. When you heat it up, all right, things start moving around faster, it dissolves a lot faster. So remember, concentration, higher the difference in concentration, the faster, the smaller the molecule, the faster. That should be relatively obvious, and the more heat or more energy you put into the system, the faster that kinetic rate's things are gonna move. All right, equilibrium is reached when there's no net movement of molecules in one direction only. All right. Molecules have a natural drive to diffuse down concentration gradients that exist between extracellular and intracellular fluids. So in your body, realize that things are going to move through passive transport. They're going to diffuse down concentration gradients. It's always going to go from higher concentration to lower concentration. That just makes sense. All right. So we're going to distribute those molecules as evenly as possible. Once they're distributed completely evenly, it should stop. Non-polar hydrophobic lipid core of plasma membranes stop diffusion and create concentration gradients by acting as selective permeability borders. So we said before that biphospholipid layer was only going to let things that were non-polar, tiny, and lipid soluble in. So everything else is not going to be able to come in. So the concentration gradient is going to be kept. So outside the cell and inside the cell, those concentration gradients should be different, all right? But if we want to actively allow them, we have to open up channels so these things can fuse back and forth, all right? Also called differentially permeable barrier, all right? So it's permeable to those three things, but differentially permeable to some of the other ones. We're gonna stop them from coming through unless we want them to come through. Right. So molecules that are able to passively diffuse through the membrane include lipid-soluble and nonpolar substances, very small molecules that can pass through the membrane or membrane channels, and this is referred to as simple diffusion. So I mentioned that it's probably 4 times now. Those are the things that can pass through simple diffusion. They don't need permission. They just automatically, naturally occur. Right. Larger nonpolar soluble or polar molecules can cross membrane, but only with assistance of carrier molecules. This is referred to as facilitated diffusion. All right, so remember, we have those carrier proteins. So we can diffuse these things back and forth across the concentration gradient, but they can't do it through simple diffusion because they're not one of those 3 things we talked about. So we have to be able to open a gate or allow them to come through. We have to provide a way across, right? Osmosis is a special name for movement of solvent, usually water, not molecules. So osmosis works with water and cell membranes. That's generally the term for it. All right, homeostatic imbalance. So, how could this clinically be relevant? All right, if the plasma membrane is severely damaged, substances diffuse freely into and out of the cell, compromising concentration gradients. Our burn patients, they lose fluids, protein, and ions that leave from damaged cells. All right, so you damage those cells, we throw off the concentration gradient, those cells usually never recover. All right, if we were talking about any kind of, if we damage a cell membrane from chemotherapeutic agents or ultraviolet light or whatever, we can throw off that selective permeable membrane. Or if we're talking about some kind of gram-positive bacteria, all right? If I can go in and we can use penicillin or some of these cephalosporins, some of these antimicrobials, I can go in and they can start breaking down that external cell wall, the nano-neg units of the gram-positive bacteria. We can allow things to go into the cell, like fluid, and cause it to blow up, or we can cause things from inside that bacterium to leak out, and then the bacterium will crenate. So we can go in and we can chemically, through antimicrobials, we can go in and alter that homeostatic balance and kill the bacteria. Right? So simple diffusion, non-polar lipid-soluble substances diffuse directly through the biphospholipid layer. All right, so these are examples of things that are non-polar and lipid-soluble. So they, like I said, these don't need permission. So oxygen, carbon dioxide, steroid hormones, all right, which you need cholesterol for. And fatty acids, because they're lipid soluble. Small amounts of very small polar substances, such as water, can even pass. But these would be very, very tiny. All right, and then if you want to see diffusion, I'm hopefully sure that you've seen this before, I hope. All right, and there's just showing you simple diffusion there. All right, so these would literally, these would move in here until these were equal. So if I had, let's say I had 50 here and 30 here, which would be a total of 80, there'd be, at some point down, there'd be 40 here and 40 there, right? All right, facilitated diffusion, certain hydrophilic molecules, hydrophobic. All right, so these are afraid of water. Glucose, amino acids, and ions. So some of these are very large, or some of them are very, very ionic, or they have charges, are transported passively down the concentration gradient so we have to have these special carrier proteins. So carrier-mediated facilitated diffusion, substances bind to the protein carriers, then we carry them across, like insulin or some of these other things. Channel-mediated facilitated diffusion, substances move through water-filled channels. So aquaporins, or these literally, these are carrier proteins. These are literally open aqueducts or channels that can move things back and forth. Carriers are transmembrane integral proteins like I showed you before. So they're going to go across the biphospholipid from extracellular fluid to intercellular fluid. They're going to have a hollow tube. They're going to allow things to go across. Each carrier transports specific polar substances. So remember, if they're nonpolar, they can go through. These are very specific polar molecules that have a very specific charge, such as sugars and amino acids, and that are too large for membrane channels. Binding of molecule, as it binds from the extracellular fluid to the molecule, specific binders, all right, causes the envelope in and change the shape that results in the molecule being moved across the membrane. So it goes and attaches, opens something up, it changes shape, it engulfs it and brings it in. Binding is limited by the number of carriers present. Right? Carriers are saturated when all are bound to molecules and are busy transporting. All right, so we only have a certain amount of carriers. Once they're saturated, we can't bring any more through. There's no more gates or doors. All right, so like I said before, if you're a non-compliant diabetic, over time that cell will start making more carriers, but at some point in time, there's no more surface area, so they can't do it. Major issue we have with non-compliant diabetics. All right, so it's going to go in. It just changes the shape. All right, as they come in, it changes shape to allow only things in. It closes it off so nothing else can attach until we empty that, and then it can go back and reconform to that shape again. All right, so passive membrane transport. All right, a few more. All right, channels with aqueous or water-filled pores are formed by transmembrane proteins. Channels transport molecules as ions or water down their concentration gradient. Specifically based on pore size or change, water channels are called aquaporins. All right, so aqua in Spanish means water, porins, so these are going to be channels. All right, so these are going to allow aqueous flow. They're filled with fluid. They're going to allow things going from extracellular to intercellular fluid, and usually it's going to be water or something with very, very tiny ions in it. All right, and there's two types. Leakage channels are always open. So we always want things to go back and forth just to keep that concentration gradient, keep things moving. So we do really have leakage channels for sodium to leak out, and then we have to actively bring potassium back in general. All right, gated channels controlled by chemical or electrical signals. All right, so we're going to have to induce these when we realize that the concentration gradient of sodium potassium is not quite even. Then we have to literally open these channels and bring these things in. All right, osmosis is a movement of solvent. Remember, osmosis is usually solvent, and let's just right now think of it as water, H2O. such as water across selectively permeable membrane, water diffused across plasma membrane through lipid bilayer. Even though water is polar, I said before, it's so small that some molecules can sneak past nonpolar lipid tails. All right, it's very, very tiny, but it is electronegative. It has a lot of electronegativity because I said before the oxygen is a huge molecule, very electronegative. Hydrogen is very, very tiny. It's got 2 small charges. So the hydrogens fill up that outer octet, all right, and make it stable, but it definitely has a charge to it. Right? Through specific water channels called aquaporins, fluid occurs, or flow occurs in water or other solvent concentration is different on the 2 sides of the membrane. So they're just going to cause equilibrium. Depending on the solute concentration, it's going to flow back and forth, making sure that solid solution is generally roughly the same. Okay. Osmolarity. All right, so this is going to be key later on. We'll start talking about some of the physiology of the human body. So osmolarity measures the concentration of total of solute particles in a solvent. All right, so the osmolarity or molarity of something depends on the actual concentration inside the solvent. So if we're talking about a monosaccharide or a disaccharide, one is single, one is double. So a disaccharide would have twice as many solute particles, so the osmolarity would be twice as big. So water concentration varies with the number of solute particles because solute particles displace water molecules. Remember, you have 100% water. The minute you start putting solutes into it, you have solute and now you have water. So there's no longer 100% concentration of water. Right? So the more solute you have, the less water it is. So the osmolarity changes. And we're talking about glucose in blood. We literally change the osmolarity of the blood with all that glucose. And if we put a whole lot of glucose in the blood, it changes the osmolarity, and then fluid comes from outside the body to try to get that osmolarity the way it should be. We'll get into that later on. So when solute concentration goes up, water concentration goes down. So remember, the more solid you add, all right, it's taking up room, so there can be less and less percentage of water. Water moves by osmosis from areas of low solute concentration to high areas of solute or low water concentration. So once again, water moves by osmosis, that free moving from areas of low solute. All right, so it has low solute, it has high concentration of water. All right. Remember, osmosis only is moving from, so figure out the water concentration. It's always going to go from area of highest water concentration to area of lowest water concentration until that is equal. All right, so if we were talking about being dehydrated, or if you were to drink salt water, all right, and your blood plasma became full of, let's say that the concentration of sodium was 7% in your blood plasma because you drank salt water. All right, so that the percentage of water in your blood would now be 93%. Right, inside of your cells or interstitial fluid, let's just say it's 0.08 or 0.8 or whatever. So it's less than 1% sodium. All right, so that has a higher water concentration, interstitial fluid of your cells. So what will happen is the fluid from your interstitial fluid of your cells will leave your interstitial fluid of your cells, go into your plasma, blood plasma, to get that osmolarity back to normal. So you're dehydrated. It's actually taking water from your body into your blood to get your blood to the right concentration. That's what causes you to be dehydrated and causes major problems. Right. When solutes of different osmolarities are separated by a membrane permeability to all the molecules. So let's just say we have 2 different solutions. Their concentrations are different. We've got a semi-permeable membrane, like kidney dialysis tubing or something. Diffusion of solutes and osmosis of water occurs across the membrane until equilibrium of solutes of water is reached. So we'll see examples of that. There'll be a video of it. So you're going to have the two solutions, all right? So figure out the water concentration of one and the water concentration of the other. So figure out what percentage of that is water. Whichever one has the highest water concentration is going to move into the area of the lower water concentration. There are no exceptions to that, right? Same concentrations of solutes and water molecules on both sides with equal volume on both sides, right? So just showing you, here is your freely permeable dialysis tubing, all right? So we know that both solutes and water molecules move down their concentration gradients. So solution with the lower osmolarity, lower solute concentration. So this literally has more water to it, percentage-wise. Solution with higher osmolarity, higher solute concentration, this has less water to it. So what's going to happen is generally things are going to move from area of higher concentration to areas of lower concentration. So when solutions of different osmolarities are separated by a membrane that's permeable only to water, only osmosis, not diffusion, will occur until equilibrium is reached. All right? All right. Water will have net movement across the membrane until osmolarity is same on both. All right? Results in high volume changes on both sides. All right? So the low solute side volume decreases. All right, the low solute is going to have the highest water concentration. The high solute side volume increases because that had least water concentration, highest solute concentration. So I said before, the water is going to go from the area of highest concentration of water to the area of lowest concentration. It's going to go from low solute into high solute. So that column, if it was permeable, it's going to actually raise. So there you see a picture of that. All right, so hydrostatic pressure is the outward pressure exerted on cell side of membranes caused by increase in volume of cells due to osmosis, also referred as back pressure. Osmotic pressure is the inward pressure due to the tendency of water to be pulled into the cell with higher osmolarities. The more solutes inside the cell, the bigger the pull of water to enter, resulting in higher osmotic pressure inside the cell. All right, so hydrostatic. All right, so think hydro, you think of water pressure. Like if you're thinking of, you know, the water tanks, if you're from the Buffalo area, you know, like in Tonawanda and Amherst, we have those big water tanks. So we have the pressure of that water pushing down. We have hydrostatic pressure, outward pressure exerted on the cell surface of membrane caused by increase in volume of cell during osmosis. Right, remember, osmosis is that water diffusion occurred to its back pressure. Right. And osmotic pressure is the inward pressure due to the tendency of water to be pulled into the cell from higher osmolarities. Right. So I'm going to see if I can get this to work. All right, so let's just say, oh, it is working. Sorry guys, it's my first time doing this. All right, let's just say we were talking about a capillary bed too. All right, so this is kind of a key component. So if this was your capillary bed, all right, and this was your heart here, not really how your heart works, but, and you know, so we had a blood pressure of 120, this is hard, over 82 or whatever. So realize, you know, that's just your blood pressure, and coming from your heart to your capillary bed, pressure is going to be higher here, it's going to be lower here. Right? So if we're talking about hydrostatic pressure, the pressure here is higher. It can also force fluid out of your capillary bed here, and you go to all the cells of your body. It's your interstitial fluid, right? Cells of your body. all right? And then your cells are going to metabolize that, and you're going to have, change this. Can I change the color? I'm going to screw this up, so I'm going to leave it alone. All right, never mind. All right, so these are your extracellular. This is the waste. All right, we want this fluid to go back. I think I just messed this all up. All right. Did I just lose it all? I did. All right, I guess we'll, all right, I guess I'll have to figure out how to do this. All right, sorry. All right, so when hydrostatic pressure equals osmotic pressure, no further net movement of water occurs. So water trying to get out equals water trying to get in. I tried to draw that with the capillary bed, but I don't know what happened. But all right, rather than make this video even longer, we'll get back to that. All right, so plant cells are surrounded by strong cells that act as to limit hydrostatic pressure levels, which in turn limit osmotic pressure. We don't have that. So plant cells will fill only so much with water and they stop. Right? And you also notice that if you don't water your plants, they get real wilted and they get real limp. There's zome and phloem columns in there. But if you water them, the turgor comes back, they rehydrate. Or if you have celery or asparagus that's been dried out, right? So if you cut the bottom of it off and you put it in a glass of water and you leave it overnight, the asparagus or celery will start pulling that fluid back up and the turgor will regenerate in those plants, even though they're technically not, alive. They'll do it. So animal cells do not have cell walls, therefore cannot limit hydrostatic or osmotic pressures. Animal cells will burst if too much water is taken in. And that will happen with, people that were, what was the contest in California? Was it for like a bike or a tickle me elmo or something weird? That their woman lost the bat and whoever could drink the most water. Well, she drank too much. She had water intoxication. Her cells literally burst, all right, because she screwed up the osmolarity. All right, outside of her cells, the concentration of sodium was less than the concentration of sodium inside the cell. So water literally had to, through diffusion, go into the cells and cause them to burst. And, it was her neural cells. So she went into a coma. Right? So water can also leave a cell causing to shrink, called crenation. Right? So tonicity is the ability of a solution to change the shape or tone of cells by altering the cell's internal water volume. All right, so isotonic solution is same. So if you don't know what that is, we'll get into the percentage. Or if you wear contacts, go look at your contact lens solution. It's isotonic. So we want that sodium concentration of the drops you're storing your contacts in, or whatever drops you're putting in your eye, to be the same as your tears. Right? So isotonic solution is the same osmolarity as inside the cell, so cell volume remains unchanged. Hyper always means over or more. Hypertonic solution has higher osmolarity than inside the cell. Remember, osmolarity has to do with the solute concentration. Right? So water flows out of the cell, resulting in cell shrinking. Shrinkage is referred to as crenation. Hypotonic solution has a lower osmolarity than inside the cell. All right, so if we're talking about sodium, that sodium concentration would be less than inside the cell. It would be less than isotonic. So water flows into the cell, resulting in cell bursting. All right. Can lead to cell bursting, called lysing. All right, and that's what happened when you're water intoxicated. You have water intoxication. You drink too much fluid. You change the osmolarity to less than isotonic. Now you have a higher concentration of water in the extracellular fluid than inside the cell. Remember, it's always going to go from high to low. So water forces, through hydrostatic pressure, gets forced into your cell, causes that cell to burst. Right? All right, so osmolarity, you know, you guys don't have to order chemicals or you're not mixing things, but, you know, if I order 6 molar hydrochloric acid or 3 molar hydrochloric acid, I want to know the concentration of that, all right? So osmolarity is equal to molarity times the number of ions or particles. Remember, ions are going to be these things from the periodic chart that have charges to them, ionic charges. So sodium chloride, table salt, 1 particle ionizes to sodium plus charge and chloride the negative charge. And you guys should remember that from your worksheets. These actually will come together to fill out that octet, to fill out that outer valence electron shell. So therefore, 1 molar solution of sodium chloride equals 2 osmolarity solution because there's 2 particles. All right, this is a classic example in every single textbook that I've ever read. Most publishers all have the same picture. All right, so this is isotonic. There is your biconcave red blood cell, your biconcave disc with all the hemoglobin to carry your oxygen and carbon dioxide. And please don't let anyone tell you that they have the same seat on the bus. All right, so your oxygen and CO2 have separate seats on that bus. They don't sit in the same sites, all right? Hypertonic solution, all right? So the solution has a higher sodium concentration than isotonic in your body. So it has a higher solution of sodium, so the water concentration is less. And remember, we said the water concentration, the highest water concentration is going to go from higher to lower. So literally what happens is water leaves your cells to get the water concentration equal on both sides. That cell will shrink, or also known as cremation. This is water intoxication. The solution is hypotonic, less than isotonic. So the sodium concentration outside the cell is less than inside the cell. Water concentration is therefore higher outside the cell. Water is going to flow in, trying to make it equal. This is when they lyse or they just burst. Okay. All right. Okay, guys, I don't even know how long that was. It felt like an hour and a half. I'm really not sure. So I am going to stop the recording. I will convert this, upload it to YouTube, and then I have to YouTube it into the PowerPoint, because if I put the Zoom file in directly, it is too big. Blackboard keeps telling me it's an invalid file. So this is what we'll need to do. So please be patient. I will post this to both lecture and lab. I'd like to go on to the other parts so we can get caught up. Please make sure that you are in mastering. So anyone who is staying on course, please jump into mastering. And if you're taking BI 151, jump in. I have assigned some stuff for chapter 3. I just need to know how it looks on your end, because even if I go into student mode, it never quite looks the same. So anyone who can do it and help me out, that'd be really, really helpful. All right, guys, it's Thursday. I think it's like 12:30. Please start studying. We're slightly behind, but I'm not worried about it because we're doing this asynchronistically, and I'll be popping up the PowerPoints for you. I know I'm long-winded and I go on and on and on, but I feel that the more you hear about it, the better, and the more examples I give you, the better. And hopefully by the time you're done with this course, you'll have an idea of how your body works. And as always, start studying.